Hello, fellow warriors. Here we are with news from the website prostatewarriors.com. Imagine a battlefield where the enemy doesn't just wear camouflage, but they literally turn off your thermal scopes.
Yeah, and they build their own secret supply lines right in the middle of a bomb strike.
Exactly. Like physically hijacking your own communication towers to turn your defenses completely against you. I mean, that is exactly what late-stage prostate cancer does.
It's terrifying, really.
It is. But today, we're taking a deep dive into the absolute cutting edge of research for you. We're going to see how science is, finally cornering this invisible enemy.
Right, and we have a very specific roadmap for our journey through the sources today. We're looking at this strictly through a timeline, starting with clinical trials first.
Makes sense. Start with what's happening now.
Exactly. We have to map out the massive advancements reaching patients right now, the present reality, before we zoom into the preclinical pipeline to see tomorrow's breakthroughs.
Yeah, it makes complete sense to start with the reality on the ground. And looking at the stack of research, the most advanced clinical data we have is the Phase 3 PROTEUS trial.
The PROTEUS trial, right.
I've seen this name floating around patient forums for a while, but I really want to understand why it's making such massive waves. We're talking about men with high-risk, localized cancer.
Right, candidates for surgery?
Yeah, candidates for surgery. And, I mean, we've been doing radical prostatectomies for decades. So what makes PROTEUS so fundamentally different?
Well, the historical approach for those high-risk cases was always pretty straightforward. You go in, you surgically remove the prostate and the pelvic lymph nodes, and you just sort of hope you got it all.
Right.
But the PROTEUS trial, which was recently published in the New England Journal of Medicine, challenges that by testing a perioperative strategy.
Perioperative, let's break that down because it's a crucial distinction.
Yeah, they took over 2,000 patients, and instead of just surgery, they added a potent hormone therapy drug called Apalutamide to standard androgen deprivation therapy.
And it doesn't just mean giving patients medicine after the surgery to clean up, right? In this trial, they gave patients 6 cycles of this combination before the operation.
Yep. 6 before, and then another 6 cycles after.
Wow. So to use a military analogy, instead of just, sitting in the ground troops and hoping they find every enemy soldier, this is like sweeping the battlefield with heavy air support for months before the troops even arrive.
That's a great way to look at it. And then keeping that air support going afterward.
To catch anyone trying to regroup.
Exactly. They hit the cancer systemically before going in with the scalpel. And the immediate pathological results, they were a massive paradigm shift.
Really? How massive?
Well, when the surgeons removed the prostate and the pathologists looked at the tissue, they looked for a pathological complete response, basically meaning they couldn't find any viable cancer cells left.
Which is the dream scenario.
Right. On standard treatment, the placebo arm, only 1.0% of patients achieved this. But with the apalutamide, it jumped to 8.9%.
Almost 9%. That's huge. But wait, why is that systemic suppression so vital in the first place?
It's because of microscopic disease. High-risk prostate cancer is just notorious for sending out microscopic scouts. Tiny clusters of cells that leave the prostate but are far too small for any MRI or PT scan to see.
And if you just remove the prostate, those scouts survive.
Exactly, and they eventually grow into metastatic tumors.
But, a better-looking slide under a microscope doesn't automatically mean the patient is cured. I mean, we've seen trials where the early pathology looked amazing, but it didn't translate to patients actually living longer.
We have, yeah. Which is exactly why the long-term data from PROTEUS is what truly matters here. This trial actually proved the hard outcome benefit.
Okay, what were the numbers?
The 5-year metastasis-free survival jumped from 73.5% on the standard arm to 78.2% with apalutamide.
Wow.
That's a 20% relative reduction in the risk of the cancer spreading or the patient dying.
That is a massive clinical victory, especially considering men with aggressive disease face up to a 50% chance of relapse within 5 years of surgery alone?
Exactly. Hitting it before those escape routes are established is crucial.
But, as any patient navigating this disease knows, prostate cancer is relentlessly adaptable. Even with early strikes, sometimes those scalps survive, they mutate, and they thrive, even when we cut off hormones.
It's a terrifying moment for a patient.
It really is. To hear your cancer is resistant. How do we fight a tumor that has already built armor against our best weapons?
Well, this brings us to the early-stage clinical data. There are striking results from a phase 1 trial for an experimental agent called ABBV-969.
ABBV-969.
Yeah, it's an antibody-drug conjugate, or ADC, designed specifically for heavily pretreated metastatic castration-resistant disease.
I'm fascinated by the mechanics of ADCs. Like, if standard chemo is carpet bombing the whole body, an ADC is more like a biological guided missile, right?
Yes, that's exactly it.
You have an antibody homing system seeking out a specific flag on the cancer cell, and it drags a highly toxic payload directly to that single target.
The homing system is everything. But what makes ABBV-969 unique is that it's a dual-target ADC. It doesn't just look for one flag. It binds to two different proteins on the surface, PSMA and STEAP1.
Oh, wow. Two targets.
Yeah, and once it locks on, it gets pulled inside the cell and releases a payload called a topoisomerase inhibitor.
Okay, let's slow down and translate that payload, because topoisomerase inhibitor is some heavy jargon. What is that actually doing inside the cell?
So think about how a cell divides. To multiply, a cancer cell has to constantly unzip and copy its DNA.
Right.
Topoisomerase is the enzyme that helps untangle the DNA strands so they can be copied smoothly. If you inhibit that enzyme, the DNA gets hopelessly knotted up.
Oh, so the copying process just catastrophically failed.
Exactly, the cell dies. And the beauty of this specific type of poison is that it completely bypasses severe peripheral neuropathy.
The permanent nerve damage in the hands and feet.
Yeah, which is so common with standard taxane chemotherapy because chemo attacks the cell's structural scaffolding instead of its DNA.
That is a massive quality of life difference for you listening. And looking at the numbers, the efficacy is staggering. We're talking a median radiographic progression-free survival of approximately 15 months.
Yes.
I mean, to put that in perspective, in this heavily pretreated population, the historical average with fallback chemo like cabazitaxel is only about 4 to 5 months. So 15 months is a giant leap.
It is a massive leap.
But I have a critical question here. The whole guided missile concept relies on the cancer waving those flags. What happens when the tumor mutates and stops expressing PSMA and STEAP1?
Yeah.
Doesn't the drug just go blind?
You've hit on one of the most difficult concepts in oncology, tumor heterogeneity.
Right.
By the time the cancer reaches this late stage, the tumor isn't just one uniform mass of identical clones, it's a diverse community. You might have a cluster of cells over here with high PSMA, while a cluster right next to it has mutated to express almost zero PSMA, but extremely high STEAP1.
That is such a daunting reality. You aren't just fighting one cancer, you're fighting multiple versions of it at the same time.
It's incredibly daunting, but that's the exact problem this drug is built to solve. By dual-targeting PSMA and STEAP1, ABBV-969 casts a much wider net.
It corners the tumor.
Exactly. The cancer now has to figure out an evolutionary pathway to drop two completely different flags simultaneously just to survive.
Which is exponentially harder.
Right. Though we should note, this potency comes with side effects. Over 40% of patients in the trial experienced grade 3 anemia, which required dose modifications or blood transfusions.
Good to know. Okay, so guided missiles are incredible for targeting physical flags. But what about the immune system? We always hear about immunotherapy working miracles for melanoma. Why can't we train our own white blood cells to do the cornering?
Well, that brings us to the phase 2 trial testing GV-100251.
Because prostate cancer is a cold tumor, right?
Historically, yes. It's notoriously resistant to standard immunotherapies. Drugs targeting the PD-1 immune checkpoint work by releasing a brake on the immune system. But in prostate cancer, releasing just that brake usually isn't enough. The immune system just wanders past the tumor.
So how is GV-100251 changing that dynamic?
It targets a newly discovered immune brake called IGSF8. In this phase 2 trial, they're combining this new IGSF8 blocker with standard anti-PD-1 therapy.
Oh, so releasing multiple brakes at once.
Exactly. Preclinical work showed blocking IGSF8 aggressively wakes up natural killer cells and T cells. Prostate cancer has several brakes holding the immune system back. Releasing multiple at the same time might force the immune system to finally attack.
It's all about stripping away the defenses. But let's look at a scenario where we actually do hit the tumor hard and it still finds a way to survive. The sources talk a lot about the radioligand therapy Pluvicto.
Right. 177-Lu PSMA 617.
Yes. 177-Lu PSMA 617. It delivers targeted beta radiation directly to PSMA-expressing cells. Highly effective. But the research shows surviving tumors find a devious escape route even after this nuclear strike.
They do. The tumor microenvironment is incredibly dynamic. When you treat with 177-Lu PSMA 617, the radiation severely damages the tumor and surrounding tissue. But the surviving cells don't just wait around to die.
Right.
The radiation creates a hypoxic environment, meaning it severely depletes the oxygen. And for the surviving cells, this triggers a literal panic button.
And what does that panic button actually do?
It forces the cancer to initiate an angiogenic rebound. The tumor starts pumping out massive amounts of chemical signals, specifically through pathways called VEGF and MET.
VEGF and MET, okay.
Yeah, and these signals force the body to rapidly build brand new blood vessels to supply the starving tumor.
So I make sure I'm picturing this correctly. The radiation bomb drops, the tumor is devastated, but the survivors hijack the body's infrastructure to secretly build new supply lines in the rubble and surround themselves with an immunosuppressive bunker.
That's a perfect description.
And this is where the phase 2 trial for a drug called Zanzalintinib comes in.
Yes. Zanzalintinib is a multi-target tyrosine kinase inhibitor. Its whole job is to shut down that specific post-radiation panic response.
How so?
Well, it blocks VEGFR2, which stops those new blood vessels from forming. It blocks the MET pathway to prevent invasion, and it blocks a specific family called the PAM kinases.
Wait, time out. VEGFR2, MET, TAM kinases, AXL, MERTK, TYR3. This is turning into a dense alphabet soup.
Fair enough, yeah.
Let's ground this for the listener. What do these TAM kinases actually do in plain English?
Right. The TAM kinases are the architects of that immunosuppressive bunker you mentioned. They recruit cells that tell the immune system to stand down and ignore the tumor.
Oh, wow.
So by blocking those specific kinases, Zanzalintinib tears down the bunker. It removes the shield.
The sequencing of this is just mind-blowing. You drop the radiation bomb, and the second the surviving cancer tries to rebuild its blood vessels and throw up shields, you hit it with Zanzalintinib to choke off the recovery.
The clinical elegance is profound. You're systematically stripping the cancer of options. First, hormones, then radiation. And when it tries to alter its microenvironment to survive the radiation, you immediately cut off those supply lines.
And doing this in chemo-naive patients, right?
Exactly, sparing them from harsh taxane drugs while delivering a devastating 1-2 punch.
But here's where the narrative takes a terrifying turn. All these strategies rely on us actually seeing the cancer. We rely on standard PSIC scans looking for that PSMA flag. What happens if the cancer just completely sheds its PSMA expression?
It vanishes from the doctor's monitors entirely.
Which is horrifying.
It's one of the fastest growing challenges in prostate oncology. The selective pressure from PSMA-targeted therapies is forcing the cancer to evolve into PSMA-negative variants or even highly aggressive neuroendocrine prostate cancer.
So it goes completely dark.
Exactly. That's why the phase 1 imaging trials in our sources are so critical. They're designing a completely new set of eyes.
Let's talk about the phase 1 trial testing CD46 ImmunoPET.
Okay, so CD46 is a protein on the cell surface acting like a biological shield against the immune system. Researchers discovered that even when prostate cancer sheds PSMA and goes invisible, it almost always keeps its CD46 shield.
Fascinating.
So they developed a radioactive tracer called 89ZR-DFO-YS5 that binds exclusively to CD46.
So it's not just thermal vision. It's like the cancer took off its brightly colored PSMA jacket to hide in a crowd. But this tracer is a UV-reactive dye that only sticks to their skin. You can track them anywhere.
That's brilliant, yes. And the results from the first in-human study of 30 men are wild. These men had cancer that was largely invisible to standard scans.
Right.
But this CD46 tracer detected 144 bone metastases compared to just 99 on a standard bone scan. And it found 33 soft tissue lesions compared to only 14 on a regular CT scan.
Finding more than double the soft tissue lesions in patients who looked clear is a massive breakthrough. But just seeing the cancer isn't the final goal, is it?
No, the ultimate objective is theranostics.
Therapy plus diagnostic.
Right. The concept is brilliantly simple. Once you prove the targeting antibody YS5 can find the CD46 protein, you just swap the payload. You take off the harmless imaging isotope and replace it with a highly lethal radioactive isotope, like actinium-225.
So you see it, then you swap the backpack for a bomb.
Literally turning the flashlight into a weapon.
And there's an early phase 1 trial in Australia doing something very similar, right?
Yeah.
Testing a tracer called DOTA STR-17126.
Yes. They're looking for a totally different target called GRPR, which is present in about a quarter of these invisible PSMA-negative tumors. They use gallium-68 to find the tumor on a PET scan.
And if it lights up?
They swapped to a low dose of 177 Lu using that exact same tracer. And if the safety profile holds up, the next step is a therapeutic trial with a lethal dose of 177 Lu.
It's all about creating a massive toolkit. When the cancer changes appearance, we switch the lens and the payload, which perfectly transitions us into the final section. We've mapped the reality right now. Now let's cross over to the preclinical frontiers.
The raw science happening in petri dishes and mouse models today.
It's exactly the fuel for tomorrow's trials.
Preclinical research is where we tear the disease apart at the cellular level. A perfect example is the experimental compound IVMT-RX4. It's hyper-focused on bone metastasis.
Because prostate cancer doesn't just passively sit in the bone. It actively invades the space.
It does. It has to aggressively communicate with the surrounding bone marrow stromal cells, tricking them into building a nest for the tumor.
Creepy.
Yeah, but IVMT-RX4 targets a protein called MDA9-syntenin, which is basically the communications array the cancer uses to orchestrate that invasion.
So the compound scrambles the communication lines. It stops the cancer from talking to the bone.
Yes. In mouse models, it drastically reduced bone metastases, but the real takeaway was combining it with docetaxel, a standard chemotherapy.
Oh, a combination approach.
Yeah, the combination improved survival far beyond either treatment alone. By blinding the tumor and preventing it from manipulating its environment, the chemotherapy became exponentially more lethal.
Speaking of lethal combinations, the final piece of research is a masterclass in drug repurposing. They found combining two existing drugs, eltanexor and zotafen, creates a fatal vulnerability in resistant prostate cancer.
It's a fascinating look at cellular infrastructure. Eltanexor blocks a protein called XPO1, which acts as the nuclear export mechanism.
Okay, nuclear export.
Right. When under immense pressure from treatments, a cancer cell sends survival blueprints and distress signals out of its nucleus. Eltanexor completely blocks that exit door.
So the blueprints are trapped.
Exactly. Meanwhile, zotafen blocks EIF4A1, a critical component of protein translation. It physically stops the cell from manufacturing new proteins.
They'll love this imagery. The tumor needs a shipping department to export blueprints and a factory to build resistance proteins. Combining these drugs locks the shipping department and shuts down the factory floor at the exact same time.
And the cancer just cannot sustain that collapse. In the lab, it drastically reduced ARV7 signaling, a notorious resistance mechanism. Without export or translation, the cancer cells triggered apoptosis. They basically realized they were broken and self-destructed.
Wow.
And researchers proved this in patient-derived organoids complex 3D models from actual human tumor tissue.
And the fact that these are repurposed drugs is huge. Taking a new molecule to a human trial takes over a decade. But eltanexor and zotafen already exist. Their safety profiles are partially understood.
It shaves years off the runway to get this into a clinical trial. And the data shows it works at relatively low doses, which gives us immense hope that it'll be well tolerated.
Incredible. So pulling this all together, we started with the PROTEUS phase 3 trial, sweeping the battlefield with apalutamide before surgery. We explored dual targeting guided missiles like ABBV-969 and Zanzalintinib, choking off blood vessels after radiation.
We saw the UV dye of CD46 and GRPR tracers spotting invisible cancer.
Right, and finally, preclinical science shutting down the cancer's microscopic shipping and manufacturing.
Looking at the big picture, we're watching a relentless microscopic arms race. Every time we introduce a therapeutic pressure, the tumor attempts a devious adaptation. But science isn't just reacting anymore, it's anticipating.
We're dual targeting, cutting off supply lines, creating thermal vision.
Exactly, which raises a provocative question for you to ponder as we close. As we map out all these escape routes, could the ultimate cure for prostate cancer rely not on a single magic bullet, but perhaps on an AI-driven, ever-shifting algorithm?
Oh, wow.
Imagine an algorithm that analyzes a patient's circulating tumor DNA, predicting the tumor's next mutation, and preemptively cornering it with a new combination before the cancer even makes its move.
A master chess match where the medicine is constantly 3 moves ahead of the disease. That is an incredibly powerful thought to leave on. To all of you listening, navigating this complex landscape, we hope this deep dive arms you with knowledge and, most importantly, hope. Stay strong and fight on.